Hello, photosynthesis fans. So back for more. That's good. So let's talk about photosynthesis today. So before we talk about photosynthesis, I wanted to talk a little bit, just for a minute or two, about the midterm. The midterm is 1 week from today, so Wednesday, March 17th. So we are going to have a midterm that's going to be similar to the first one in terms of structure. You may notice there's a mitosis, meiosis question thrown in there. So we'll talk about that. We're going to get to mitosis and meiosis very soon. That will be the next topic. And we should have that all covered by the end of the week. And Monday will be a review session, and then Wednesday will be the midterm. So like before, I'm going to try to have a sample midterm for you and some sample multiple choice questions online. I'm working on that right now. Hopefully I'll have those up by Friday. Shouldn't be a problem for me to get those up by Friday. And so you have something to look at over the weekend, because the review session won't be till Monday. So you're going to have to do a little bit of review ahead of time. So just a reminder, we covered 6 topics, or we're going to cover 6 topics, 7 all the way through topic 12. And something that you can do for me before Monday that would probably be very helpful, besides studying, is basically reviewing all the reactions. So if you think about what we've covered glycolysis, we covered pyruvate oxidation, covered the citric acid cycle, oxidative phosphorylation, fermentation, and that is all cellular respiration. So what I'd recommend for these reactions, that you basically figure out what they are, right? Where they happen, and also basically what the reactants and what the products are. For photosynthesis, we had 2 reactions. We had the light-dependent reactions and the Calvin cycle. That we're going to talk about the Calvin cycle today. So like I said, just some quick hints on the midterm. Like I said, I'm going to get that review material posted by Friday, so you can look at it over the weekend. So big warning that these topics here are quite technical. There's a lot of words, pyruvate, acetyl-CoA, a lot of things going on. And if you're looking for an area to focus, these topics here all have are about 2 lectures each. 8 and 9 are kind of short. Topic 7 was a little bit of a carryover from topics 1 to 6, but had some kind of overlapping ideas in terms of energy and structures and things like that. But 8 and 9 were sort of try to get you ready for topics 10 and 11. But definitely the focus will be 7, 10, 11, and 12. And like I said, I want you to try to review these reactions as you get ready for the midterm. So let's get back to photosynthesis and talk about the Calvin cycle. So if you remember last day, we talked about the light reactions. So since we've talked about the light reactions, you should be in good shape for understanding tomorrow's lab. The light reactions, of course, involve light and water, and the water gets split and makes oxygen, and the light and the electrons from the water end up being put onto these molecules here, ATP and NADPH. So today we're going to talk about how we're going to take those high energy bonds from ATP and NADPH. We're going to take those high energy electrons, we're going to grab a little bit of carbon dioxide, and ultimately we're going to make some sugar. In the end, we're going to recycle our NADPH and our ADP and PI. So let's take a look at the Calvin cycle. So I'll tell you one thing, I love the Calvin cycle, but maybe not that it's the most interesting chemistry, but I love Calvin cycle because of its name, and I'll get to that in a second. So first, let's talk about what the Calvin cycle is. So I couldn't resist putting a little bit of Darth Vader in there. So the Calvin cycle is probably the most common name for this thing. And that's what our textbook is using. But it goes by a whole bunch of different names. Sometimes people call it the dark reactions. I don't really like the dark reactions because they don't happen in the dark. They just don't need light. So sometimes they're called the light independent reactions. Sometimes these other guys are given some credit, Benson and Bassam. These are all, Calvin was a scientist, Benson, Bassam, they're all scientists that kind of contributed to this whole thing. Calvin gets the most credit for it. And sometimes we're calling it carbon fixation, which means we take carbon dioxide and we're going to basically fix it into organic compounds. So I'll tell you why I like the Calvin cycle, because I think Calvin's name is funny. Not Calvin just in general, but let me ask you this. If you were Calvin's mom and you have a bouncing baby boy, what would be the worst possible name you could call Calvin? Well, I'll tell you, or at least in my opinion, is that Melvin does not go with the name if your last name is Calvin. So that's what I think that's funny about the Calvin cycle, because yeah, it's just a great name, or silly or something, and I'm just glad I'm not named Melvin Calvin. But anyway, great guy, I'm sure. He major contributions to science, and basically what he was doing was studying photosynthesis by using radioactive carbon dioxide. Yeah, I know people are thinking radioactive carbon dioxide does not sound like fun, but it's what he did. There are some pictures of him doing it. He basically grew up some algae, and when your compounds are radioactive, they're a little bit easier to trace when you're isolating compounds. And so that's what he was doing, and he derived this whole thing that we now know as the Calvin cycle. So let's take a look at some of the details here. I don't have it in many details as the citric acid cycle, but a few details in there are worth mentioning. So the first thing is, of course, we have our carbon dioxide. And we have this compound here, RUBP. So it's a big name. It's ribulose 1,5-bisphosphate. So this is a carbohydrate, ribulose. Hopefully that's the clue. Anytime you see ose in something, it's a carbohydrate. It has 2 phosphates on it. So this thing has phosphates, which means it's actually already high energy, and it's been kind of charged from the previous round of the cycle. So what we're going to do is take an enzyme, and this enzyme here, you can see it has a big fancy name, ribulose 1,5-bisphosphate. You can see it has the substrate in the name, and the enzyme is a carboxylase. So that means it's going to do something with carbon dioxide. So we can call it affectionately Rubisco. And I have some pictures of Rubisco. And what that's going to do is basically make this, we're going to take this 5-carbon compound, RUBP, and we're going to add a carbon dioxide, and we're going to make this short-lived 6-carbon compound. I'm not sure why it's short-lived. It doesn't really matter. But what we're going to end up with is this 3-phosphoglycerate. So we've done it. We've captured carbon, but it's not done yet. We've got a whole cycle to talk about. So let's talk about Rubisco. I remember a few years ago, I had mentioned Rubisco, and I found out that Rubisco is an enzyme, and it is also apparently a wrapper in Toronto. It turns out one of the students I had, he was involved in something I had no idea was even a thing. He does these rap battles. I don't know if you guys know what rap battles are. That's where I guess 2 people rap and they insult each other. Anyway, this is a thing. That's what he did. He's from Toronto, and he knew a guy there named Rubisco. So anyway, it's a wrapper. It's also maybe the most important enzyme on the planet, because this thing is taking carbon dioxide out of the atmosphere, locking it into organic compounds, which we as animals are making use of. So there it is. You can see it's actually found, it's a membrane-bound protein. I'm not actually sure if that's in the thylakoid membranes or not. It doesn't really matter. Most of the Calvin cycle takes place in the stroma. So here is kind of like a skeleton of the Calvin cycle. You can see it's showing the number of carbons. So we have 5 carbons here, and then there's Rubisco, and then now we have 6 carbons. That breaks in half, and we have 3. These 3 carbon things, so 3-phosphoglycerate. So now we have to add some more energy to the system because we're trying to build something up. So remember those high energy electrons? We had a bunch of ATP we made in the light reactions. We had a bunch of NADPH. Both of these have high energy electrons found in bonds, so potential energy. and that gets built up to this thing here, glyceraldehyde 3-phosphate. So this I want you to remember, or at least remember G3p, okay? Why is G3p important? This is the product of photosynthesis. So I know a lot of you have been trained to think of glucose as the product of photosynthesis, but it's not. Glucose is just sometimes what plants make. Some plants don't make glucose. Some plants make fructose. Some plants make sucrose. They all use G3p. So G3p, I had pointed out, was part of the glycolysis pathway. And so it's pretty easy to take 2G3p and make a glucose out of it, by the way. And you can see down here, it says glucose and other compounds. So that's kind of part of it. We built it up, we made some sugar, and the last part of this cycle is just kind of regenerating that RUBP. So some of the G3ps end up making glucose and other compounds, and some of them go back and regenerate that RUBP, and that takes a little bit more ATP. So you can see this is pretty energetically expensive, right? So we're talking about, what is that, 9 ATP there and 6 NADPH, and each NADPH is equivalent to 2.5 ATP. So, I mean, this is, I don't know the exact number without doing the calculation, but it takes quite a bit to make one G3p molecule. And but that's a good thing, because we're trying to store energy in organic compounds. We're trying to build things. We're trying to build cells and cell parts, and this is what the plant is trying to do. So there's the Calvin cycle in a nutshell. Couple of things to remember. Remember the ingredients are carbon dioxide, ATP, and NADPH. I don't care about the stoichiometry, we're not going to count. And of course, we create the broken down product, so the ADP and inorganic phosphate, and the broken down NADPH, which is NADP+. The final product is glyceraldehyde 3-phosphate. So that's kind of the big thing to know about the Calvin cycle. And the last thing to remember is the name of that enzyme, and you can call it the abbreviation. You do not have to call it ribulose 1,5-bisphosphate carboxylase. You can remember Rubisco, and I'm fine with that. So I have this little review thing that I usually do, and what I'm going to do today is actually just put this off till Monday. Going to review photosynthesis. I want to kind of review the light reactions and the Calvin cycle once people have had a time to digest. I know photosynthesis it's a lot, right? It takes time to digest. You can have to review it a couple of times to kind of understand some of the basics behind it. And this one here, once you've had time to review, I want to kind of compare and contrast a few things around photosynthesis and respiration. And like I said, I'm going to put that off till Monday's review session. So if there's anything you need to know, like I said, one slide is this picture. You want to add a little bit more to it, we can throw in this this tree. You know, where does the water come from? Hopefully you know where water comes from in plants. It comes from the soil and it goes up through the roots and ends up in the leaves. So like I said, remember this slide here because it shows the whole thing, the light reactions out in the Calvin cycle. And if you need to memorize some details, memorize that picture there that shows those main ingredients of what is going in and what is coming out of the light reactions and Calvin cycle, and of course how they're intertwined. You know, I look at this picture here and I can imagine, I don't know how many types of multiple choice questions I could ask you. You know, what are the products of the light reactions? Or what is the purpose of the light reactions? So the purpose of the light reactions is to produce ATP and NADPH for the Calvin cycle, right? Don't say the purpose of the light reactions is to create oxygen. Oxygen has no purpose in photosynthesis. It is a useful compound. We love it, but it's not the purpose of photosynthesis. It's just a kind of a happy side effect. So there are a couple other things I wanted to talk about regarding photosynthesis. These are just repeats of the slides I showed you. A couple other things about photosynthesis in terms of what is going on. I think something I mentioned before was that, you know, photosynthesis or chloroplasts in general are found in the green part of a plant. So that's just something good to note, right? Is that not every part of a plant is green. Not every part of a plant is chloroplast. But you probably know that the part that's usually almost always green are the leaves. And so this is a leaf cross section, and you can kind of see what's going on here. And we have these little pores. And these pores in the leaf are actually for gas exchange. So you can see carbon dioxide is going in and oxygen is coming out. And usually they lose a little bit of water vapor as well. Now these pores are called stoma. So do not get that confused. Remember, we also talked about a stroma, and now we're talking about a stoma. So the stroma, remember that is part of, that's the kind of like the cytoplasm of the chloroplast. It's the innermost compartment, or it's the inner compartment where the Calvin cycle occurs. Stoma is the pore in a leaf, or if you have many of them, you can call them stomata. So you have an idea, there's just the words here. I know there was one year where I actually asked part of my midterm, I said, compare, what are these two things? And you know, some people got the two of them mixed up. So keep in mind, you know, you need to know your vocabulary. I'll show you a couple other pictures of these things. These stoma are surrounded by these two cells called guard cells. And these guard cells can basically close or open the pore in some species of plants. So you can imagine that's really handy on a hot, dry day. The plant can close its stomata in order to prevent drying out. The disadvantage is they can't exchange gas, and so photosynthesis slows down. There are mechanisms in some plants for opening them during the night and storing carbon dioxide, and then closing them during the day when it's hot and dry to prevent drying out. There are mechanisms for that, and you can read about that in the textbook if you're interested in this kind of thing. But there's some pictures. You can see these cells are not totally useless as well. They do have their chloroplasts in them as well. There's some other images of them. You can see this one. The chloroplasts are fluorescing in a nice red color. Kind of look like eyeballs or the eye of Sauron or something like that. So there's some more stomata. And I have this little video for you of photosynthesis. And I'm going to leave this for you to play on your own time, because what I want to do instead is do a Kahoot. And so load up your app, and what I'm going to do is play the Kahoot for you. What did I do with it? There it is. And I'm going to warn you, this one is a little bit more technical. And hopefully it's going to encourage you to study this stuff here. So load up your app. We'll play this for you, and it'll hopefully help us to review some of these things too. There we go. There's the pin. 8, 9, 7, 4, 3. Okay, here we go. What is the name of the sugar formed during photosynthesis? So we just covered that. So I see a couple of people had sucrose, which is not wrong. A lot of plants do make sucrose, but the common product in all plants is glyceraldehyde 3-phosphate. Which of the following is a product of photosynthesis? Oxygen, good job. Carbon dioxide is not a product of photosynthesis. Carbon dioxide is a reactant. What is the name of the pigment that absorbs sunlight to power photosynthesis? Chlorophyll. Chloroplasts are the organelle that contains chlorophyll. All right, watch out, Mark. Emma's coming in hot. Question 4, which part of photosynthesis does not require light? Hint, this is the thing we just talked about. So Calvin cycle is correct. Citric acid cycle is part of respiration. Question 5, where does photosystem 2 take an electron? So here's a hint, photosystem 2 is the first part of the light reactions, right at the very beginning. So tough one there. I guess I don't have any image to show you. Maybe the question is worded poorly. Again, this is a Kahoot I kind of borrowed and adapted from somebody else. Remember that the light comes in photosynthesis, it hits chlorophyll, and the electrons from chlorophyll are replenished from electrons from water. So maybe just a poorly worded question there. Question 6, which part of a plant cell contains the light-absorbing molecules that start photosynthesis? So where is the chlorophyll found? Chlorophyll is found in the thylakoids. So that is the innermost membrane, the green part of the chloroplast. Question 7, the products of photosynthesis are the reactants for which reaction? Hopefully that one's an easy one. Correct answer is cellular respiration. So products of photosynthesis, generally we're thinking of glucose and oxygen, and that's what's required for respiration. Couple more. Which carrier, so we're talking electron carrier, provides the high energy electrons needed to make the carbon-hydrogen bonds during Calvin cycle? So again, I don't know if I like this question as much as I thought it would have been, because ATP is also correct, but ATP isn't usually considered an electron carrier. So it's kind of a misleading question. Again, this is the problem when you borrow questions and don't read them carefully, but ATP is sort of okay, but NADPH is the most correct answer. NADH is found in respiration, but not photosynthesis. Okay, good job, Tristan. In which location does the Calvin cycle take place? It is in the stroma. So not the stoma, but the stroma with the R. Okay, one more question. Let's see if you can hold on to that lead, Tristan. Which products of the light reaction are used for the Calvin cycle? Okay, looks like people did okay. ATP and NADPH. So carbon dioxide is not a product of the light reaction, and water is not a product of the light reaction. Those are both ingredients of various parts of photosynthesis, but not products. All right, let's see how we made out. In third place, Nina, bronze medal. Silver goes to Mark. And the gold, I'm open for Tristan. She snuck in there at the end. Well done. Well done. 8 out of 10. Very good. So as I warned you, that one is a little harder. You really have to know lots about photosynthesis to do well in that one. And like I said, it's going to involve a little bit of studying. So another thing I'll try to remember to do is share the link for this Kahoot collection so you can look over these questions again. Maybe if I get a chance, I'll try to review some of these questions and I'm just taking a look here. I'll review some of these questions on the Kahoot and see if I can fix them up. There's just another image from a different textbook showing photosynthesis and how it's corresponding with respiration. And you can see it's sort of have all those molecules in there that we've been talking about that's in there. I guess the one difference here is they're calling the citric acid cycle the Krebs cycle, but I'm okay with either one of those terms. Okay, so there's a reminder to do the Kahoot and a nice rainbow for you because, hey, rainbows are nice. All right, so basically right where I want to be today is I wanted to finish photosynthesis and start on the next topic. So let me just share that with you here. And we are going to start talking about the cell cycle. So by the cell cycle, I mean we're going to talk about mitosis and meiosis. So usually people find this relatively straightforward, at least the mitosis part. Some people get a little bit confused with the meiosis part, but if you're familiar with it, great. And it should be an easy unit and relatively light compared to the respiration photosynthesis stuff. Topic 12 is also, because we're going to talk about DNA, it's sort of a good segue into the last part of the course, which is all about DNA. So let's get into this and talk about DNA. So one thing to think about is, why do cells need to reproduce? Part of it is just reproduction, particularly for a unicellular organism. This is how you divide and make babies, right? So if you take a look, there's an amoeba on the left, and it's dividing, and now there's going to be 2 amoeba. If you are big organisms like us, growth and development, right? We all start off as that one fertilized cell, and you know, we get bigger and eventually become larger animals. We start off as little people and become bigger people. And maybe the other obvious one is just general tissue renewal and repair. Our skin cells are constantly dividing because, well, that's how skin works. We lose old cells and we replace them with new ones. So just a reminder, we did talk about cell division a little bit already. We talked about cell division in bacteria. And remember, the term for that is binary fission. So binary fission, one thing to point out is that we have 1 chromosome. That's it. When it replicates, there's 2. So 2 copies of that 1 chromosome. And the way it divides is you can see that the chromosome there is attached to the cell membrane. So as this bacterial cell basically elongates, this chromosome here on the right gets pulled to the right, chromosome here on the left gets pulled over to the left. No spindle fibers, nothing fancy. There's no nucleus to worry about. It's a relatively simple process. Eventually, you end up getting the pinching that happens here during the division, and that's where the cell wall and cell membrane form a new one in between, and then you end up with 2 daughter cells. In most eukaryotes, you get mitosis and meiosis. There are a few kind of other types of cell division that are similar to mitosis and meiosis. They have different names, but usually we're looking at mitosis and meiosis. And in this case here, we're involving, look at this. This cell here has how many chromosomes? It looks like 8. Separating out multiple chromosomes, you've got a nucleus to worry about. We make these spindle fibers here, and it's a little bit more of a complex process. So that's what we're going to be talking about today, at least mitosis. We're not going to get to meiosis today. So quick test yourself question, just a thinking question, right? This one says, for a protist, what is the advantage of using a eukaryotic like mitosis rather than a prokaryotic binary fission? So it's kind of asking, why do mitosis in the first place? So let's take a look at these answers here. Binary fission does not allow for the formation of new organelles. No, not even close. Has nothing to do with organelles forming, right? Mitosis allows for the orderly and efficient segregation of multiple linear chromosomes. That sounds like a good one. I'm going to put a check mark there and just read the other ones just to make sure. Mitosis is faster than binary fission. Definitely not. Binary fission can be 20 minutes in some cells. Mitosis, we're looking at hours. For human cell, it's like 16 to 24 hours. D, mitosis allows for lower rates of error per chromosome replication. No, replication is a totally different process, so definitely not. E, binary fission does not allow the organism to have complex cells. I'm not sure if that is right or not. That's kind of a weird statement. So definitely the correct answer is B, is that we are dealing with multiple chromosomes, and it's very important that we separate them efficiently and orderly. We don't want to mess up. We don't want to end up with 2 copies of chromosome 3 and no copies of chromosome 4. We want to have everything all good and organized. So before we talk about mitosis and meiosis, I want to talk a little bit about chromatin and DNA packaging in general. So I think we had defined chromatin before, and I had mentioned that that chromatin and chromosomes, that the names are kind of used interchangeably. And these things basically mean DNA and associated proteins. So sometimes, like I said, these words are used relatively interchangeably. In most cases, when we say chromatin, we kind of mean loosely packed, and chromosomes often we mean it's kind of more tightly packed. But both words are used interchangeably. So don't get too caught up in that. I am going to give you some more definitions though in a couple of minutes here. So I have a little video here I want to show you, and then I'm going to do a diagram for you. So this is, I don't know how they do it. It's kind of an animated chalkboard. And they're going to show you a little bit about how DNA is packaged in a cell. So I'll play that for you right now. Just got to make sure I'm sharing the computer volume. All right, here goes.
In this video, we will be zooming in on the chromosomes in a cell, and then unpacking the chromosomes in order to see the cell's DNA. We will look at a neuron, but we could look at another cell type as well, since DNA is present in cells throughout the body. The chromosomes are located in the nucleus of the cell. Let's isolate a single chromosome. DNA is tightly wound up in a chromosome. We need to unravel the chromosome into what is called chromatin fiber in order to access the DNA for this neuron. Let's now unpack the chromatin fiber. Chromatin is made up of DNA that is tightly wrapped around proteins known as histones. DNA contains 4 sorts of nucleotide bases: thymine, adenine, guanine, and cytosine.
So he doesn't do an amazing job of going into the details, and that's what I'm going to do right now, is make a diagram for you and kind of just dissect a little bit about what's going on with the chromosome packing. So I'm just going to share this whiteboard, and you can see I have a title there. It says DNA packaging in eukaryotic cells. So let's draw some DNA, and I'm going to make some notes around this kind of thing. So first of all, if you think about DNA, DNA is basically a double helix. So there's my double helix. So DNA double helix. So basically, that double helix is completely just nucleotides. And so after that, what happens is it gets spooled around histone proteins. So I'm going to draw histone protein for you. Let's see here, I'll do that in red. And they showed that in the video. So there's my histone protein. And my DNA, it kind of, let me see here, how, if I can do this. Here's my DNA. Kind of spools around it, sort of like this. Something like that. So these are, okay, so DNA spooled around, I'll even change my font color for you, histone proteins. So these histone proteins are really essential for packaging this DNA. You can imagine you've got this skinny, crazy long strand. We're talking about some chromosomes, I think the largest chromosome is more than 1 meter long if you were to unspool all that DNA, and we're trying to package into a cell that's like 50 micrometers, right? So hugely important. So the next step is we can actually pack those histone proteins together into something called a nucleosome. I'm going to try to draw a nucleosome for you here. If you don't like my drawing, at least I'll let you know there's 8 histones. So we got 1, 2, 3, 4, and I'll sort of try to figure out if I can draw the ones in the back. Maybe the ones in the back, I'll shade them in like that. 1, 2, 3, 4. Okay, so I'm a wonderful artist. I'm sure you love my artwork. There's my DNA. I'm just going to wrap my DNA around it like that. I know that's amazing. So when you get 8 histones, this is called a nucleosome. So that's 8 histones, and that's about 200 base pairs. 8 histones, I'll say stacked histones. So that's about 200 base pairs. I think it could be 196. I can't quite remember the exact number. So I'm just going to scroll up here. Hopefully everyone got everything up at the top there. So these nucleosomes, they basically end up in something called these 30 nanometer chromatin fibers. So I'll draw that for you. So what you end up with is these 30 nanometer chromatin fibers, and I'm going to kind of draw it like that. And the nucleosomes are kind of in there like that. So you don't have to be an amazing artist to be a scientist. So this is 30 nanometer chromatin fibers. So I'll show you some actual pictures from the textbook in a couple of minutes here. This is about 6 nucleosomes per turn, but I'm not even going to write that down. You can imagine these are 30 nanometers. So if you think in nanometers, that's about how big we're looking at here. Okay, so the next thing here is we have something called looped domains. So let's me draw some looped domains for you. A loop domain kind of looks like this. So these are called looped domains. And this is actually what we are going to call Euchromatin. Hold on a second, I can't spell obviously. I gotta get an eraser. There we go. Euchromatin. So what do I mean by U? Remember, U for eukaryotic means true. So this stuff here is, I'm just trying to think of the word. This is, we'll call it loosely packed. Loosely packed DNA and associated proteins. DNA plus, I'm just going to write proteins rather than associated proteins. And those proteins are mostly histones. There are a few other proteins in there that help to package DNA, but I'm not going to go through those since the main ones are the histone proteins. So this is usually what people say is chromatin. We can go a little bit further in the organization of DNA, and we can have some condensation as well that happens. So the condensation, again, like I said, I'm just kind of trying to draw this. If you kind of picture those loop domains, and then they kind of get squished or scrunched together like this. So there's my condensation, and you can see that, you know, I'm making this 4 loops again, and they're even packed tighter. So condensation. And this condensation, often we call this heterochromatin. So this stuff here is tightly packed DNA. Tightly packed DNA plus proteins. So one more step here for you is that this usually gets, when it gets condensed, basically we end up in something that we would call a chromosome or a metaphase chromosome. So that's going to look something like this, right? And that familiar kind of X shape. So this is a metaphase chromosome. Although you will see these in prophase as well. But they sort of start condensing in prophase, so they're complete by the time they get to metaphase. Okay, so there's the diagram for you. And it looks like my handwriting got a little bit bigger as I went along. And I'll show you what we have in the textbook and some actual images of this DNA. So back to the PowerPoint, I just got to be able to find it. There it is. And so here's images from the textbook. You can see there's my, my double helix here on the left. We've got our histone proteins here, and histone proteins, they stack into these nucleosomes. The nucleosomes are, like I said, we've got 8 histones, and that's about 200 base pairs. And I guess what I didn't show in the fibers is that they coil up a little bit. And these here are the 30 nanometer chromatin fibers, which is going to be shown on the next slide. Maybe the next slide. This is just kind of a zoom in on the histones and their packing. You can see the histones are all given names. We have H2, AA, and H2B, and 3 and 4. Don't know where H1 comes in. Not sure why it's missing. It's probably associated somehow, just not as part of the nucleosomes. You can see on the right, there's actually some electron micrographs. Actually, this is an electron micrograph of some nucleosomes. And this one here is actually using some probe microscopy, atomic force microscopy, to show the nucleosomes. So they're quite visible, which is why they get a nice name, because if you can see it, somebody looking under a microscope is going to name it. Here's the 30 nanometer chromatin fibers. So like I said, it's about 6 nucleosomes per turn, and then they form these loop domains. And this is my euchromatin. As it condenses and you get your chromosome, we start to call it heterochromatin. Excuse me, a little frog in my throat today. So bottom line is, in the end, we are compacting that DNA molecule about 1/10,000th of its original length, which is really quite incredible, going from like a meter to like, 10 micrometers, which is like, that's just incredible. So here's a picture of a cell. This is a transmission electron micrograph, and you can see this is the nucleus actually. If you look outside here, we've got some mitochondria out there, it looks like. A little bit of endoplasmic reticulum kind of showing at the edge there. This big thing in the middle is the nucleolus. And if you take a look, there's another one down there at the bottom and another nucleolus. Animal cells usually have 1 or 2. Plant cells can have more, depending on the plant. And you can see we have this sort of diffuse stuff here. And this is the euchromatin. This is the less condensed, loosely packed stuff. It contains the histone proteins, it's just not condensed. So this is the DNA the cell has access to and is going to use, the genes that are going to access. The other chromatin here is the heterochromatin. It usually shows up darker, it's kind of more packed, and any given cell is going to have a little bit of both in there, unless it's undergoing mitosis, where it's all going to be packed up, ready to go to be for cell division, because it's going to be trying to divide, it's not trying to make new products. So some terminology. This is worth it to kind of try to talk about what is going on here in terms of the terminology. I know sometimes there's a little bit of confusion about what's going on here. So let's just take a look and talk about this, and I'll try to squeeze in some definitions as we do it, right? So I've already defined what a chromosome is for you. It's a piece of DNA. So a piece of DNA plus associated proteins. Okay, so that's what we're looking at here. And it looks like that particular chromosome is condensed, and that's doesn't really matter. What we're trying to show you is it all together, and I'm not going to draw a big strandy thing. So that chromosome during interphase is going to replicate. So that means you're going to get 2 copies. And when you get 2 copies, they pair up together like this. And we call those sister chromatids. So sister chromatids, definition would be 2 replicated chromosomes. Sorry, 2 replicated, I'm going to say copies. Oh, I can't, my eraser's not working. Okay, I'll just scratch that out. 2 replicated copies of a chromosome. And I could add that have not yet separated or something like that prior to separation. But basically 2 replicated copies, so they're identical. And those are called sister chromatids. So what is the centromere? You can see the centromere is labeled here. It's somewhere in the middle. The centromere, this is the attachment point of the spindle fibers. The attachment site of spindle fibers on a chromosome. or sister chromatids. On sister chromatids. Okay, and one more word for you. So by the way, the centromere is a DNA sequence. Sorry about that, I meant to squeeze that in there. I didn't plan this slide out well enough to leave enough space for my definitions. So let me just see if I can separate these out, make them a little easier to read. So last thing is kinetochore. What is that? So this has the word kinetic in it. And these are actually proteins. And these are actually motor proteins. Motor proteins. And what do these motor proteins do? Well, remember, motor proteins move things. And what are we moving? We are moving chromosomes. So they move chromosomes or sister chromatids. And they move them along the spindle fibers. So there's some definitions of some things we're going to talk about. We're going to use all that terminology, okay? And when we get to meiosis, we're going to throw in some more terminology as well. But we don't have to worry about meiosis today. We're going to save that one for Friday. So let's take a look. There's a picture of a fluorescence micrograph, fluorescence microscopy, and you can see that they have the different things labeled. The spindle fibers are these things in green. The DNA is this blue kind of shadowy stuff. And the kinetochores are not made of DNA, they're made of proteins, and they stain a different color. They stain in a pinkish color. So let me just see where we are with time. Okay, a few minutes left. Another definition for you, something you may hear about when you talk about genetics, is sometimes people talk about a karyotype. Karyotype is basically a preparation of a chromosome set of an organism. So this used to be a really common thing people did back in the early genetics days, because you could take a cell under, you'd find it in metaphase when all the chromosomes were kind of all condensed, and very carefully the technician would stain them and separate out the chromosomes, and you could count how many chromosomes are in an organism. So this one, for example, this is a human karyotype. You can see there's actually 23 pairs. So 46 chromosomes, and these arrows, I don't know what happened to them. They were supposed to be here and here. I was just showing that this person is a male. They have an X and a Y. And so you can take a look at those things. You can see different banding pattern, and people would talk about, chromosome 8 and a certain region. And it's kind of useful for just classifying where genes are and those kind of things. I wanted to show you this one here. We can use fluorescence, of course, nowadays. The other one was black and white. But I wanted to show you here what is going on during metaphase. Remember, these things are replicated, and we actually have sister chromatids is what we're looking at here. And kind of like that cartoon that shows that, because it's not so obvious in the actual karyotype. So karyotypes can be used to learn a little bit about the genetics of organism, sometimes determine the sex of an organism. You probably know how mammals work when it comes to sex. Mammals, if you have two X's, you're usually a female, and if you have an X and a Y, you're usually a male. There are a few biological exceptions to that. There are some females with 3 X's, and there are some females with XY and some males with XX, but those aren't as common. The usual circumstance is XX is female, XY is male. But if you're not a mammal, there are lots of other interesting and weird things that go on out there. So you can see, for example, in some insects and worms, if you are a female, it means you have 2 X's. And you're a male if you just get 1 X. So it's a little bit of a different system and mechanism of determining the sexes between these organisms. If you're birds, we don't even use X and Y, we call them Z and W for some reason, because again, it's a little bit of a different system. If you're an alligator, you're not concerned about which chromosomes you have. It actually has a lot more to do with the incubation conditions when you were an egg. So there's a lot of different ways that this can be done, because hey, it's biology, and there's lots of cool things out there. So quick test yourself question, and I'm going to wrap up in a minute here. So this one says the centromere is a region which centrosomes migrate to during mitosis. Nope. The nucleus is located prior to mitosis. Nope. Chromatids remain attached to one another until anaphase. Yes, that is the correct answer. This is where these things are joined together and where the spindle fibers attach. So I just wanted to talk a little bit about interphase, and then I'll let you go for today. So this is showing the cell cycle. And notice that mitosis is just a little tiny part of that cell cycle. So the whole cell cycle, by the way, is in a human cell is something like 16 to 24 hours, depending on the cell type. And mitosis itself is about 1 hour of that whole thing. So what is going on? We have this thing in the middle, which is interphase. And interphase was named by mitosis researchers, so they call it an interruption to mitosis, but it's really the main event. And what we have here are some different stages. You can see we have G1. So G1, the official name for G1 is gap 1, but a lot of textbooks are starting to call it growth 1 now as well. Gap meaning it's a gap between mitosis events. You can imagine what's going on there. The cell is growing, it's collecting resources, it's getting bigger. It's, you can't just divide all the time, you got to grow in between. S phase is for DNA synthesis, and you can imagine what's going on there. The DNA is replicating. And then G2 is a little bit more growth. And then we're going to have mitosis. Mitosis, by the way, is the division of the nucleus. And we also have cytokinesis, which is the division of the cytoplasm. So usually they kind of happen at the same time, but again, this is biology and things don't have to do it all the same time, all the same way every time. Sometimes there are exceptions to that. So next day, we're going to talk about the stages of mitosis, and then we're going to talk about meiosis. So we've got our prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis, and we'll go through the steps of that. And I'm hoping it's going to be relatively familiar territory for everybody. And like I said, it gets a little tricky when we get to meiosis, so we'll get there, and that will all be on Friday. All right, so thanks for coming out today. Lab tomorrow will be on photosynthesis, and I hope that goes well for you. And I will see you when I see you.